Welcome back to Chapter 9. In the second and final video, we're going to look at the types of receptors that we have to bind to signaling molecules. We're going to see that some of these receptors are on the membrane or the surface of the cell, but sometimes these receptors are actually in the cytoplasm of the cell, and that's true if the signaling molecule is hydrophobic or water-fearing and can pass through the cell membrane. So let's look at cell surface receptors first. If my signaling molecule is hydrophilic or water-loving, let's say it's over here, that's my signaling molecule, that means it cannot pass through the cell membrane. Remember, the cell membrane is made of phospholipids, and only small non-polar hydrophobic molecules can pass. So if you have some kind of hydrophilic water-loving signaling molecule, since it can't get through the cell membrane, it has to bind to receptors that are embedded on the cell membrane. So the signaling molecule could bind to this and cause some kind of intracellular effect. For these, usually what happens is you get a fast response in terms of whatever happens inside the cell will occur pretty quickly, but these are also usually short-term effects from seconds to maybe minutes. It's not something that would cause a long-term days or years type of effect. Another term for these signaling molecules are ligands. So this little red dot that I drew that represents the signaling molecule, we can also call that a ligand. These are chemical signals. And there are a lot of different types. Some of them are small proteins, some of them are ions, for example, and we'll see several across the semester. For the types of receptors they bind to, we're going to look at three types. One is an ion channel linked receptor, another is called a G protein linked receptor, and finally we have enzyme linked receptors, and we'll go through each of these one at a time. Let's start with the ion channel linked receptor. Remember, in this case, the ligand or the signaling molecule cannot cross the cell membrane, so it has to bind to the membrane bound receptor. And in this case, it looks like when it binds the receptor, it causes the receptor to change shape, and the receptor happens to be an ion channel. So when it changes shape, it opens up, and these ions, little green spheres, are now able to pass into the cell and cause some kind of intracellular effect. When the ligand is released from the receptor, the channel closes, and now no more ions can cross. So this is actually a really nice example of what happens when our neurons tell our muscles to move. So if you want to move your arm, for example, if you are lifting a dumbbell, this is my hand, and I want to lift a dumbbell, your brain will send a signal through a neuron, and the neuron will release signals onto your muscle. And the muscle cells have a channel that looks just like this. So when the neurotransmitter binds to it, the channel opens up, or the receptor opens up, sodium rushes in and causes a series of events inside the cell that allows for muscle contraction. And once you're done, the signals come off or get broken down, get degraded, the channels close, and then the muscle relaxes. A second really common example of a membrane-bound receptor are the G protein-linked receptors. Here's what it looks like. Here is the receptor. And again, since your signaling molecule or ligand cannot cross the cell membrane, it will bind to the G protein linked receptor. So this is called a G protein linked receptor because the receptor in green is linked to a G protein. And there are three subunits to the G protein, an alpha, beta, and gamma subunit. So all three of those pieces make up the G protein. It looks like when the receptor is not active, or the G protein is not active, it's bound to GDP, which is a lot like, or very similar to ADP, adenosine diphosphate. In this case, we have guanosine diphosphate. But once the signal molecule binds, it looks like what happens is the G protein becomes activated. And instead of GDP, which comes off, GTP becomes bound to the G protein complex. Upon binding, the G protein is activated, and the alpha subunit will separate from the beta and gamma subunits, and that will cause some kind of intracellular response. Eventually, if the signaling molecule comes off, or if other steps occur and GTP is hydrolyzed and becomes GDP again, then the process stops. So we have the gamma and beta subunits reassociated, and everything can happen again if the next ligand or signaling molecule binds once more. And this is a bit beyond the scope of our class, but if you're interested in what kind of ways we use G protein link receptors, one example is in the back of the eye where your retina is. We have cells called photoreceptors, and one type of photoreceptor are rod cells. These rod cells, when light is shined in the eye, or if your eye detects light, it actually will activate G protein coupled receptors that allow your brain to respond to the light or detect the light. Our third most common example of membrane-bound receptors are our enzyme-linked receptors, and more specifically, our tyrosine kinase receptors. This one's a bit different from the others. So usually what happens is you need two signaling molecules to activate the process. So I see one there, and now here I have two. Before the tyrosine kinase receptor is activated, you can see that there are two components or subunits to the TK receptor, TK. Once both signaling molecules bind, one binds to each of these components, then the receptor forms a dimer. This is called dimerization. So a dimer is formed. Why did I write it? It's right there. So this dimer is formed and it becomes activated. So once dimerization occurs and it becomes activated, it's interesting that each side will phosphorylate the other side. They phosphorylate one another and they take the phosphates from ATP. Once both sides are phosphorylated, it's fully activated and causes other cellular responses. Again, you don't have to memorize this part, but one example of how we use tyrosine kinase receptors in our bodies is to respond to insulin. So insulin is a hormone that's released from the pancreas after a meal. So you just ate a bunch of food, maybe you just ate dinner. Insulin will be high in the blood, and ultimately insulin will bind to tyrosine kinase receptors on cells, especially in your liver and your muscles, and that will trigger a series of events that ultimately allow glucose transporters to be inserted into the cell membranes. Now, since you just ate a meal, you'll have higher blood glucose levels than normal. Once these glucose receptors are embedded in the membrane, now you can take in the glucose and lower your glucose levels in the blood back to normal. The common response of these tyrosine kinase receptors in certain cells is to cause cell division. Unfortunately, in some types of cancer, especially in breast cancer, it's pretty well studied, these tyrosine kinase receptors are called HER2 receptors, HER2, which stands for Human Epidermal Growth Factor Receptor number 2. In some types of breast cancer, they're either overexpressed, causing the cell to divide uncontrollably, or these tyrosine kinase receptors are mutated so that they're permanently on, they're permanently activated, which also causes unregulated cell division. Here are some vocabulary terms that are important and related to the cell signaling process. The first is something called signal transduction, and taken literally, signal transduction or transduction refers to a change in the type of energy that is received or transmitted. And it kind of makes sense here because if I have a cell and if my signal, let's say this is my signaling molecule, cannot get into the cell directly and it has to do or transmit a signal through binding to a surface membrane receptor, then this is called signal transduction because it's transmitting a signal indirectly. It's telling this receptor to do other things, step 1, step 2, step 3 inside the cell. Another term that's important is dimerization, and we saw this in the tyrosine kinase receptors, when two receptors bind to each other to form a stable or activated complex. Everything that happens after the signaling molecule binds to the receptor, so all of these events or a cascade of events that happen inside the cell, that is all part of the signaling pathway. And then sometimes we have signal integration, where a single signaling molecule can actually activate two or more different receptors or receptor types to ultimately cause the same response in the cell. And one example of this is epinephrine. Epinephrine, also known as adrenaline, think about what happens in your body when adrenaline is released. So usually it's like someone jumps out and scares you, or something happens where your heart starts racing, you're breathing fast, you're trying to get away from a dangerous situation. Epinephrine is released and causes that immediate response. So to cause that really, really quick response, epinephrine or adrenaline actually bind to several types of surface receptors, which ultimately activate the same response, increasing heart rate, increasing blood flow, increasing respiratory rate, etc. Now let's switch gears for a second. We've been looking at receptors on the surface of the membrane for signals that cannot cross the cell membrane. But what if we have hydrophobic signaling molecules that do have the ability to cross that phospholipid bilayer? So we're going to look here at hydrophobic signaling molecules. Since they can cross the cell membrane, they don't have to have membrane-bound receptors, like the picture shown here. Instead, their signal receptors can actually be inside the cell in the cytoplasm, which is most commonly the case for these hydrophobic signaling molecules or ligands. In this case, the signaling molecule, if it's hydrophobic, usually has some kind of carrier protein to transport it through the blood, because the blood is mostly water. And then the carrier protein drops off the hydrophobic signaling molecule, it crosses through the cell membrane, and now it binds to the intracellular receptor. Ultimately, what happens with these kinds of hormones or signaling molecules is they pass through the nuclear envelope, and they ultimately bind to the DNA and change our gene expression. So this usually means they will activate the transcription and translation of certain genes, which we haven't learned yet, which means that we're going to basically read our DNA and ultimately make new proteins. So these kinds of signaling molecules usually take longer. It takes a longer amount of time before the cell responds because gene expression takes some time, but the effects are generally long-term. Usually you'll see cell growth or tissue growth over time. Examples of these hydrophobic ligands include steroid hormones, and steroid hormones are generally derived from cholesterol and include examples like estradiol, which is an estrogen derivative, and testosterone. So these kinds of hormones can cross the cell membrane, bind to their internal receptors, those intracellular receptors in the cytoplasm, and again, usually change gene expression, for example, increasing cell or tissue growth. One example of a hydrophobic ligand that acts a little bit differently from the other hydrophobic ligands is nitric oxide. Nitric oxide is a gas, but it also acts as a ligand. It can cross the cell membrane, but unlike other steroid hormones or these steroid hormones, nitric oxide has an immediate effect. And it's often used to treat heart disease in the form of nitroglycerin. It's also used in Viagra to treat erectile dysfunction. So how it works in both cases, interestingly, is it dilates blood vessels very quickly, increasing blood flow in that area, either the heart or in the penis. So what are the things that can happen inside of the cell besides gene expression when a signaling molecule binds to its receptor? We're going to take a step back and go back to the types of signaling molecules that cannot cross the cell membrane. So instead they bind to some kind of membrane-bound receptor and cause some kind of intracellular reaction. So some of these include phosphorylation. So after something like a G protein is activated or the tyrosine kinase is activated, the next steps could include activation of an enzyme called a kinase, And remember, kinases phosphorylate things. They can transfer phosphate groups between different molecules. And phosphorylation often has the effect of turning things on or off inside the cell. So often if I have an enzyme, let's say this circle thing is an enzyme, and usually the state of it is off, it's inactive. If I add a phosphate group to it, often it becomes active or on. Sometimes it's the opposite, where phosphorylation turns it off and dephosphorylation turns it on, but more commonly phosphorylating something will turn it on. Something else that can happen in the cell is the production of molecules called second messengers. So these are called second messengers because the original messenger, that signaling molecule or ligand, cannot cross the membrane. So it activates the production of second messengers that are inside the cell. And one really common example of a second messenger is something called cyclic AMP. And this is produced by an enzyme called adenylyl cyclase. If this enzyme, adenylyl cyclase, is activated, it takes ATP and turns it into a ring shape structure. So you can see two of the phosphate groups come off, and you form a cyclic version of adenosine monophosphate. So this is cyclic AMP. So again, cyclic AMP is called a second messenger because the original or primary or first messenger was that signaling molecule that could not cross into the cell. So it binds to the receptor. Here's the G protein example. A G protein is activated, and a really common pathway is the G protein will next activate adenylyl cyclase to produce cyclic AMP, and then the cyclic AMP will cause activation of a kinase that causes phosphorylation and activation of downstream proteins. So we see this as a process, or that term earlier, I said signal transduction. Since the signal cannot get into the cell, the pathway is activated indirectly. So a bunch of things happen inside the cell that is a result of the signaling molecule changing the shape of the receptor on that cell membrane. Besides cyclic AMP, two additional pretty common examples of second messengers include DAG, diacylglycerol, and IP3, inositol triphosphate. These are created by an enzyme called phospholipase C. And what usually happens to produce or activate phospholipase C is that some kind of G protein activated pathway will activate phospholipase C. Phospholipase C will then take PIP2, which is derived from the phospholipids from your cell membrane. And you don't have to know this, but PIP2 stands for phosphatidyl inositol bisphosphate. So don't worry about that. PIP2 from the membrane is broken down by phospholipase C, producing these two second messengers. And again, they usually have some kind of effect, such as activating downstream kinases, phosphorylating things inside the cell, and turning on or off other proteins inside of the cell. And that takes us to the end of chapter 9. If you're watching the videos in order, the next chapter would be chapter 10, which looks at cell reproduction and mitosis. All right, thanks for listening.